All right, we're going to get started in just a couple of minutes. If you haven't already, okay. So people are just getting settled. We're going to kick it off in just a minute. Thank you. All right. Victor, has it cleared out there? Everybody's back in? Yeah. Perfect. All right, we'll get started then. So thank you all for coming to what is our second in our new series of Dairy Innovation Hub faculty seminars. Before we get to our speaker today, I just want to remind of a few logistics. We do have colleagues joining us from River Falls and Platteville and Madison virtually. So if you ask a question later, make sure to press and hold your microphone button and speak into the microphone. And that's why I'm standing here. As you all know, I don't need a microphone except for when we're streaming. So it is a great excitement of the hub that we have been able to hire 14 faculty already across the 3 campuses. And we're in the midst of a second round of hiring. So in addition to the 5 faculty we've already hired at Madison, we're in the process of hiring a few more. So it's been very exciting for all of us to add new colleagues and new critical mass to campus and to our colleagues' campuses at Platteville and River Falls. So with all of these new incoming people, one thing we wanted to do was be able to feature them, what their great innovative ideas are, help them meet potential collaborators on the 3 campuses, and so that's where this idea came from. So appreciate you coming out today. We do plan on this being a monthly thing. We'll take summer recess here as we all go to, take summers off. I mean, get all of our research done. No. This is when we cram all the research in, right, in the summer months. So we'll not have seminars over the summer, but we will reconvene them this fall. So with that said, and without any further ado, I'm pleased to introduce our speaker today, Dr. Joe Pierre. He's an assistant professor in the Department of Nutritional Sciences, focusing on the links between milk components and human health, leading to new dairy products for individuals across the human life cycle and age cycle, consistent with personalized nutrition. Prior to joining UW, Joe was an assistant professor affiliated with the divisions of Neonatology and Pediatric Obesity in the Department of Pediatrics at the University of Tennessee College of Medicine. Joe is also an alumni of UW-Madison, so we are very pleased to have him back here in Wisconsin. Please welcome Joe Pierre.
Thank you, Heather, and hello to Platteville and River Falls and everyone watching online. I'd first like to just give a thanks to Heather for being so welcoming and encouraging during this transition, and to my department chair, Dave Eide, over the last 6 months. You guys have been fantastic, so thank you. I appreciate that. So what I would like to do today, just give you a brief background, where I came from, what I've done. And then I'm not going to talk about the last 10 years of research or so, but more so forward-looking on some of our ideas and things that we're currently working on in the lab. So, as mentioned, I'm new to the faculty, but I'm not new to campus. I came here in 2005 to run track, which was kind of fun, but you can see from our faces it's not all that much fun. But coming to UW gave me an opportunity to get involved in undergraduate research in the School of Medicine and in CALS. And so that led to pursuing a degree in biology and natural science, and then sticking around and examining some research topics in nutritional science that are related to today's topics, including surgical nutrition. From there, I was very fortunate to do a postdoc fellowship at the University of Chicago under Eugene Chang in gastroenterology, hepatology, and nutrition, and started to build a repertoire of different biomedical research skill sets in gastroenterology, inflammatory bowel disease, thinking about bariatric surgery, mechanisms of microbial contribution and bile acids and how metabolic set points are altered. And then from there, again, I was fortunate. My lab 1.0 was at the University of Tennessee in the departments mentioned. And this was it was a great place. I was there for 5 years. We had a lot of success, and I enjoyed the Memphis weather and barbecue. But was very fortunate in October to come to this great research institution, a place that I admire, and very excited to be establishing my lab 2.0. And so this is a picture from our old lab, and Talia. Let's see, Talia's in the back hiding up there, but she came with me from Memphis, so she's our lab manager. We're recruiting another scientist. We have a position open for a postdoc, and we have a number of great undergrads who have come in and helped us with a lot of the ongoing projects and started to pitch in with our new ideas. And we also have a recruit, Karen Antunes, coming to us from Brazil, an outstanding student. She was a nutritional scientist and then spent 4 or 5 years as an RD. And she's interested in studying dairy in a couple of the projects that I'll be talking about too. And I need to submit the application to get that position. Anyway, so she's coming soon, and I'll get that to you. So more formalities. Our mission is discovery. We approach our research topics through trying to find things that maybe haven't been revealed before. And we do this within the realm of host diet and microbe interactions with metabolic outcomes. And the goal is to take these discoveries and ideally come up with new therapeutic targets to get after underlying mechanisms of diseases. And we do this also through a diverse community of ideas and solutions with the eventual vision of improving human health. So in our lab, we rely heavily on preclinical models, murine dietary, surgical models, bariatric surgery, parenteral nutrition. We've done some intestinal resection studies in addition to diet. We also use sequencing approaches, either amplicon or whole genome, to better define and characterize the microbiomes in our studies. Anaerobic and aerobic culturing techniques to pull out bugs of interest, and that also is important for our ability to do germ-free work, because if we want to collect a sample from a human or a mouse, we need to keep those microbes viable. So we use an anaerobic chamber, and we can then successfully transplant those communities to look at causality. We work closely with clinical partners, Department of Surgery and Gastroenterology here. We'll talk a little bit about one of the collaborators in our bariatric surgery study. And then this position uniquely allows this opportunity to go to the agricultural source and samples, the CDR, food science, animal science, and be able to take milk samples all the way through from farm to bedside in improving human health. So this is a very exciting opportunity for me. And so why is milk fascinating? Why would I be interested in this position? Well, reading through and learning about milk, I get more excited every day. I'm thinking about domestication, 3 to 4 separate bovine domestication events in history, at least 10,000 years ago, giving us this enormous dietary advantage. You have complete protein sources, you have fat during famine, you have mineral and vitamin sources, and possibly as far back as 8,000 years ago, you start to see lactase selection in certain human populations. So we're certainly relying on these sources and being able to take them as we grow out of adolescence. And with oligosaccharides, it provides a consistent prebiotic source. There's also immunological advantages. So the human FC alpha receptor and the bovine FC gamma 2R have a common gene origin, a common ancestor. And it turns out that a lot of the bovine immunoglobulins will bind to human FC receptors and modulate T cell, B cell, macrophage function, dendritic sites. And this is important in the gut, thinking about gut-associated lymphoid tissue programming and education. So there's interesting questions here. And milk, of course, as a mammal, thinking about the importance of appropriate gut microbial assemblage and support, and how each of these things all of these things. They're not shown in proportion, but just an example of all of the different nutritional components in a milk sample. And they each have a role. So this is colostrum. It could be applied to milk as well, thinking about how the importance of these various components in a lot of very relevant processes in the body. Immune modulation, cell growth, antimicrobial functions, in addition to being nutrients. And so in early life and in many other cases throughout life, we think about microbial succession on a landscape. So the gut, you're born from a sterile environment and you begin to assemble microbes. And you can think about this after a fire or on a deserted island. You start to grow grasses and then bushes and trees and then animals move in. And so this ecology, this increasing complexity of the ecology can be paralleled to what happens in the gut. And so this is what you see going from birth through adolescence and eventually coming off of milk and being able to take solid foods. The milk diet is very important for this founder microbiome population. And so we possibly can harness the components that have certain effects in the microbiome to maximize the benefits in other scenarios in life. These are just a graph of colostrum to transitional to mature milk, and I think this is actually from humans. But you see that a lot of these things are found very, very high in concentration and begin to drop off into the mature milk, but they're still present. And other things such as the sugar increases. And the effect of these individual immune modulatory things such as, here's SIGA, but in bovine milk, IgG, and lactoferrin, is to keep certain microbes under control while the lactose and the oligosaccharides foster lactobacilluses and bifidobacteria, which are very important founder microbes, and begin to make a stable functional gut microbiome that is beneficial to the host. So, covered a lot of different background concepts, and so in thinking about milk and what we want to do, there's a lot to pick from. There's a lot of different directions we can go, and I'm sure as we get into our research and talk with more colleagues on campus, we'll come up with dozens of potential project and topics that we can go after. The three that we're currently working on are related to lipid fractions, GMP, a kappa casein fraction, and thinking about immunoglobulins, which are not new to research and have been explored since the 1960s, but we'd like to take another look ourselves in thinking about what we might be able to use them for outside of the box. So these are the three project or topic areas: exploring the targets and applications of IgG, some optimization and opportunities in surgical nutrition care, and then long-term outcomes in bariatric surgery. And we'll just discuss some of our ideas there. So number one, exploring the targets and new applications. When I gave a talk at the DIH summit back in, I think it was October, November, I showed this slide, the concept of collecting these antibodies and how they target, it's known that they target certain microbes and not others, and what does that mean for someone consuming these things. So you have this broad immune repertoire from cow to cow, farm to farm, strain of cattle, the season that are shaping the immune repertoire, and that's transferred from the cow and through the mammary gland into the milk. And then many of these antibodies detect human-relevant pathogens or other microbes, and they can be detected and used by human cells. And also these antibodies directly alter epithelial cell biology. When they're sensed, glycosylation patterns change. And so it's just an interesting thing. The infectious disease researchers, especially third world diarrheal illnesses, researchers have used these and really been successful in preventing diarrheal illness. But there's plenty of other applications. So a new lab getting into this area, what we've done is we brought 34 strains of bacteria with us, And then Bob Kirby and Federico Ray, we went through their freezers and looked at old stocks and pellets and came up with this broad array of microbes, largely many of them human-derived, that cover many phyla, family, and taxonomies of the human gut microbiome to come up with 70 bacteria, and then we also have some fungi and proteases that are important in gut disease that we could start with to think about how do we screen these. So the thing that we're working on right now, we have all of the strains in the lab, is generating a dot blot. So you just take a nitrocellulose membrane, we heat shock the microbes, and we can blot them out, knowing the grid pattern and what dot is what. We can expose different milk samples to the dot blot, and I forgot to mention, I call it BISA, Bovine Immunoglobulin Screening Assay, and when IgGs are present in a particular milk sample specific to one of these microbes, we can see that through a fluorescent detection using an anti-bovine IgG with FITC. So very conceptual. We got different commercial colostrum sources. We looked at 2% milk from Babcock, and we found in thinking about IgG, it's present in milligram quantities in colostrum, and it's very, very low, 50, 70 micrograms in 2% milk. So maybe we should go to raw milk, mature milk in between there, estimated around 500 micrograms per mil. And so this is just an illustration of kind of what this might look like. Many of these microbes were selected, the most obvious because of association with infection or inflammatory bowel disease, but also associated with or against obesity, associated with or against breast cancers or certain cancers that we have other funding on, and clustering these on the BISA panel. So this is an example outcome. We have a standard curve, we have a positive control, which is basically an anti-IgG antibody to make sure that we're detecting it. And then we, working with Jessica Cedarquist, are thinking about what types of samples should we be collecting. First calf or eighth calf, the age of the animal, previous infections or drug use, pharmacological interventions, diet. If you can think of others, let me know. We're brainstorming on this as we're just beginning our sample collection to see if we find something interesting. So our goal is to get 50 or 100 milk samples just to start. And we'll treat these dot plots as a continuous biological variable, so we can do Spearman rank correlation coefficients and come up with different classifications shown in this dendrogram just as an example. It may be that certain milks are very low in diversity of the IgG compartment and others are very high. It might be that we can classify them by common microbes in their specificity, or it could completely be wrong, maybe it won't work. But this is a very quick and relatively cheap thing to survey, and so we're excited about getting started. So what would we do if we had these samples? What would be the next step? You have this donor cow, and this one's really high in diversity, for example, and this one's very low. So what? So the next step is to do immunoglobulin fractionation through protein G agarose, and this will be on our key samples. And that's just to purify the antibody. And then we can take human or other animal microbiome samples and expose it to the purified bovine IgG from our source, and we can do sorting, microbial sorting based on IgG positive versus negative communities. And just as a first pass of causality, we can look at what do the microbes that are targeted, the microbes that are targeted versus untargeted have on a host. Another thing that's a much more natural experiment, since germ-free animals are not in the wild, is to just feed the purified bovine IgGs to wild-type SPF mice and then look at the effects on the gut physiology and microbiome, just to begin exploring this. And if anyone has questions, I know we're supposed to maybe wait till the end, but if something's unclear, just let me know. So the mechanisms, it's well recognized that These antibodies can opsonize bacteria, creating some selection pressure that they can potentially inform the gut-associated lymphoid tissue through the Peyer's patch, and then the generation of host immunoglobulins, and then this epithelial programming, which has been recently demonstrated. Let's see, I can't read the title from here. Immunoglobulin G from bovine milk primes intestinal epithelial cells for increased bifidocolonization, a beneficial microbe. So infectious disease is old news. Not that it's not useful there, but a lot of interest there. I've been talking with some of my colleagues, one that studies a genetic form of enteral colitis called Hirschsprung's disease, and when I told him about this project, he said, we need to test it. We need to test BISA, or test these antibodies in my patient population of Hirschsprung's. It's a 1 in 5,000 birth of live births. Others would be, since we are beginning to understand some of the mechanisms in ileal Crohn's disease, there's various targets and proteases that we know are there that may be driving flares. Could we make targeted milks that contain antibodies to neutralize these types of things? From a broader perspective, metabolic augmentation. So thinking back to if we just feed a purified IgG from different sources, what happens to metabolism? And then modulating enteral formulations. You have patients that can't eat on their own and they require a tube into the stomach. As far as I can tell, looking at baby formulas and enteral feeds, they don't contain antibodies. They have developed oligosaccharides and other fractions of milk, but not the immunoglobulins. And then talking with Vanessa Leone, the ability to enrich bifido would be useful for some of her NAFLD work. So maybe there's an opportunity there, and we're discussing a pilot for that. This is just rehashing the IBD thing, and since I have so many slides, I'm going to skip over these. Blue sky goal of developing targeted either colostrum or milk or medical foods that might reduce the incidences of flares. IBD has no cure. It's this recurring chronic disease, and something like this would be amazing. The second is applications in surgical nutrition. And so here, I'm going to introduce surgical nutrition. If we're not familiar with some of the terms, and talk a little bit, just a little bit about this. I can't talk about it too much for reasons I'll describe. So parenteral nutrition is intravenous nutrition when a patient can't eat at all. Maybe they've had trauma surgery, general surgery, intestinal resection. If there's a obstruction of the intestine, so you have to take elemental feeds, glucose, amino acids, vitamins, minerals, electrolytes, and then lipid emulsions, and put them directly into circulation to make sure the patient doesn't starve or develop malnutrition. And so this is parenteral nutrition. Enteral nutrition is just a feeding tube, a tracheostomy tube, or a gastrostomy tube, or any other version of this where you're putting a complex diet directly into the GI tract. So focusing on parenteral nutrition, the great thing is that it saves lives. 30,000 Americans are permanently on this, 300,000 adults and babies at some point during an inpatient stay in the hospital will receive parenteral nutrition. And it prevents malnutrition, it supports metabolism, but it has infectious risks. It has hepatic complications, largely related to bile acid signaling that may be disrupted by phytosterols, because a lot of the lipids that are used are derived from soy, olive oil. In Europe, they use some fish oils. And then there's, in a neonatal setting, there's non-optimal organ development, so stunting of the brain. The GI tract is not being fed normally, so you have a lack of incretin release, and altered mucosal immune responses. All right, so in the lab, we can place these catheters either into the jugular or the stomach, and so we can model this. And we spent a couple of years looking at humans and trying to look at, these are neonates in Tennessee, what was associated with what. So hepatic organ stress in the setting of TPN was associated with shifts in primary to secondary bile acids. So these are bile acid species that are modulated by the gut microbiome. And so we wanted to test the importance of the liver's ability to test secondary bile acids by making a knockout mouse. And I'm just kind of moving through this because there's a lot of material. What we found was that when the animal wasn't able to sense bile acids, they developed larger livers, elevated AST and ALT as markers of hepatic stress. The circulating bile acids were elevated. Let's see. The composition of that elevated bile acid pool was dramatically shifted and associated with hepatic stress, so increased macrophages, Kupffer cell activation, IL-6, and so the liver was not happy. And we kind of came up with this graphical abstract that certain bile acids and the ability to sense certain bile acids are very important under parenteral nutrition to prevent cholestasis. Another more recent study that we're working on is our neonatal model. So there's no neonatal model in a rodent of TPN, and this is where most of the clinical problems remain. Adults, we've kind of figured it out, we can manage them. But babies have developmental problems, long-term metabolic chronic disease risks, again, brain stunting. And so the ability to model this age of life would be powerful. We can also do a second surgery to remove a catheter and put pups back onto regular foods and begin to understand the long-term effects of a period of parenteral nutrition. Through the behavioral analysis core here on campus, you can imagine doing a study where you feed mice for 5 days, which is equivalent of maybe a little under a year in their lifespan, converted to, or maybe 6 months of a human comparison, and then put them onto a Western diet or a low-fat diet and examine how do these organs adapt back to function in adolescence and development to get after long-term development and metabolism questions. So rambling there, sorry. But what we did was we fed parenteral nutrition to 10-day-old pups. This is developmentally equivalent to a newborn human. But we first either gave antibiotics or water to the dams because a lot of the children that are critically ill and receive parenteral nutrition are also on antibiotics. So we wanted to create a dysbiosis ahead of time. And so we've done this study. We're in the process of preparing it for publication. This is a very, very, very busy slide. It was very late at night when I decided to just drop this in, but we have a number of things that we looked at. So changes in intestinal length, villus height, these would be expected. We surveyed the spleen, the pancreas, the liver. Are there changes that we should be looking for as we're trying to understand the phenotype? One thing that we did find was that, and this is the slide, or this is the data that's not on the slide, that the liver had a very pronounced pro-inflammatory response with parenteral nutrition by itself. And that injury, that pro-inflammatory injury, was entirely blocked when we gave antibiotics with parenteral nutrition. But in the process, the antibiotics messed up bile acid metabolism, and specifically by driving up CYP27A1 and inhibiting, to some extent, 7A1 and 8B1. So the classical versus alternative bile acid pathways are markedly different in gene expression, and then the output, the composite here, was that we're augmenting which bile acids are being made. And so we're writing this up to begin to set the stage for what are the metabolic implications and how might metabolic programming be changed by these early life exposures. Talia, up in the back, is interested in this phenotype. We found that parenteral nutrition compared with enteral nutrition inhibits brain growth, but that antibiotics in either case had also inhibited, so antibiotics on EN had the most significant stunting. And she's begun looking at myelination patterns. I didn't have comparisons or raw data here to show you. She's a neuroscience major, so this piqued her curiosity. All right, so we talked about a lot of things related to liver dysfunction and programming. And one of the current problems, I have to be careful here, John. So hepatic complications, phytosterols, the sources of TPN maybe are not great. And then they don't appear, the lipids that are currently used don't appear to support brain growth. And this is a major problem. So we think we have a solution to this. Of course, WARF won't let us talk about it, so this is collaboration with Grace Lewis, John Lucy, Nathan Price, and the pharmacist at UW. So I hope to talk about this next time. And I'm sure everyone will remember all of the background slides leading up to why this is important. The other application is enteral nutrition, and this is very easy to do in the mouse. You just put a gastrostomy tube through into the stomach and put them in a metabolic cage, and we can provide controlled nutrition. This is just another idea, might be revisiting the value of these antibodies, whether they have direct opsonization effects, whether they may enhance the body's own immune system in a more beneficial way, whether they may reprogram epithelial cell biology to enhance quote-unquote good bacteria. So this is one thing that we could easily test with some of the sources that we identify in the BISA screen. There's some discussion of this in the literature already. Is there a role for enteral administration of these immunoglobulins in pediatric care? I can't find, this is a review, and there's opinions on it, but this is something that we could test. And this is just showing that in premature birth, the child would normally still be accumulating IgGs, and then in the human we shift over to SIGA. But these are readily, the IgGs are readily isolatable in bovine sources. The other, as mentioned earlier in the lecture, is this observation that IgG may enhance bifido colonization, and so Vanessa and I, we've talked about this potential idea in her cholesterol and NAFLD study. All right, finally, long-term outcomes in bariatric surgery. How are we doing on time?
You're good.
Good?
Yep.
Okay. And I have in quotations, perhaps in the perimenopausal state. So westernized diseases, obesity, diabetes, certain cancers have been rising over the last 50 years, and this is an outdated map just illustrating the, those trends from the CDC. And in the most morbidly obese individuals, the most effective therapy is bariatric surgery. And so you can see on the left the different styles of surgery, either gastric banding, Roux-en-Y, there's duodenal switches or vertical sleeve. It turns out vertical sleeve is the fastest, has low complication rates, and hospitals can still bill. So they have switched to this over the last 10 to 15 years. These different styles of surgery have variable long-term outcomes, based on what surgery was done, based on how well the patient adhered to dietary counseling and management. And so this is another model that we have in the lab. This is just unpublished data. We were interested in some of the drugs that patients are fed or provided around the time of surgery, in this case fluconazole, which is an antifungal. And we found that over the 6 weeks following surgery that animals that were previously exposed for 10 days to fluconazole regained all their weight, and a lot of this was in the fat compartment. And so we spent a couple of years exploring this, and it turned out just to be a disruption of some of the CYP genes, and we still haven't published it, but some of the things that we can do in the lab then is looking at body composition, looking at adipose tissues, the quality of the liver. We can put them into total energy expenditure, caging, metabolic cages, and explore all of these things. Another observation that we made, I think 3 years ago, was that bariatric surgery, in addition to losing all of the adipose tissue soon after surgery, unfortunately patients also lose skeletal mass and bone mass. and this might be related to malabsorption, but also changes in catabolism. And so in this particular study, we found that even though the lean mass was recovered, that the strength of the animals was not. So the grip strength was changed. There weren't changes in autophagy-related genes, but rather protein catabolism was altered, and some other things here that we would like to repeat in the setting of post-surgical whey protein. And so there's a lot to read in the literature. I'm trying to grasp the different fractions of whey proteins and working with Dr. Ney to come up with good ideas and approaches here. But the idea is that whey protein fractions could be provided in the setting of bariatric surgery. Of course, you want to lose fat and maintain fat loss so that the patient in practice wouldn't have to come back for a second surgery. But you're also thinking about protecting that protein and muscle compartment and protecting the bone. So there's a lot of literature out there on whey protein. There's some, thanks to Dr. Ney and others over the last 15 to 20 years, looking at GMP and looking at weight gain, adiposity, tissue, body composition, and some of the data are not consistent between animal models and humans, and so really trying to tease out what's going on here. A study by Dr. Ney looking at bone phenotype in mice after GMP, and one study I could find with whey protein supplementation enhancing body fat and weight loss in women long after bariatric surgery, randomized control. But there's nothing on PubMed, not a single study that looks at GMP after bariatric surgery. So we're discussing this. We have mice in-house that we're aging, and where I think we're going to go is to examine the effect of GMP on long-term success after bariatric surgery. This is just a review showing all of the different potential beneficial effects. I'll highlight remineralization, the prebiotic effect, and modulation of digestion and metabolism. So this is a study that Karen Antunes, the new graduate student, will focus on. The premise is that GMP may have these beneficial metabolic effects, and they may be more pronounced later in life, especially in the peri and postmenopausal state. There's effects on satiety, but also on sustained fat loss. They may preserve bone and muscle. And this is testable. So we're going to do 2 cohorts in the mouse model to explore this at about 9 months of age in the mouse. So this is perimenopausal. And Dr. Funk, who's the bariatric surgeon at UW, is excited about this idea because he's looking for things, interventions and therapies that might improve his patient's well-being and long-term success. So we have 130 female mice. We're taking these 2 cohorts out to 6 months and then put them onto a 12 weeks of high-fat diet to induce diet-induced obesity. Then we'll do the VSG surgery at around 9 months of age. And still under construction, but conceptually, we're going to have GMP and various control diets. And we'll follow up over 8 weeks with a whole battery of readouts, looking at these, the utility of using GMP and other whey products for metabolic benefit. All right, so that's what I have for ideas. I kind of stumbled through all of it, but I'd like to acknowledge the current lab, the people in the lab, the former members, Dr. Ray and Bob Kirby, collaborators in DIH. Everyone has been extremely willing to engage and collaborate and jump on new project ideas. Other collaborators at UW, Dr. Ney, Luke Funk, Vanessa Leone, and the pharmacist at UW, Joe Smith. Grant support, and to end, a picture of Lake Mendota. So from there, I'll go to Heather.
Thank you. Questions for Joe?
was that clear?
Especially that one slide.
Yeah, there's a lot of stuff. Yes?
Joe, with the bovine,
I
regarding the bovine IgA and IgG, so I'm assuming they make it through the acid stomach fine, but could you talk a little bit about how in the process of digestion these antibodies could be modified and how this could affect, or is it known?
Yeah, so there, I could find about 10 sources that looked at the survival of IgGs throughout the GI tract. So if a patient had lower stomach acid, or if they were collecting from an ileostomy tube, the IgG would survive somewhere between 40 to 70%. Other studies suggest it's much lower, maybe 5-10%. And then in babies, maybe because of a lack of stomach acidity, again, higher. So it has been explored. I don't know. The acidity and protease exposure would probably cleave some components of the conserved and variable region.
But it would be easy to protect it if needed. I'm just thinking that could create a lot of variation if, like, one mouse or one rat or one human degraded it more than another. It wasn't you could really increase your variation in your readouts because it wasn't reaching the colon.
Yeah. Well, another approach might be to use the IBD capsules that are specifically designed to break open at the ileal pH, and they do that with infliximab or whatever one of the NSAID, the very strong NSAIDs. They break them open in the distal GI. So you could do that. Okay.
All right, sounds like I'm on. So I have a couple of questions. Kind of a loaded question, but I'm just curious to share your thoughts about the opportunity to work within the Dairy Innovation Hub, like what that kind of means to you in design, in terms of your research connectivity, collaborations, and things like that. So that's one, just curious what your opinions are. The other question is, so GMP is kind of a unique peptide, right? And so what's like next on the horizon in that general realm of surveilling dairy components for bioactive fractions or components? So those two questions. Thank you.
Okay. So I'll start with the second one. There's the lactalbumins, and it appears in the literature there's interest in comparing the alpha and beta, and especially in formulas, that the amino acid profile of maybe I think that the alpha lactalbumin may be beneficial for growth, brain development, neurotransmitter synthesis. So that's just one that I'm aware of that people are beginning to work on. Next generation GMP, I would have to lean on Dr. Ney for a thorough answer, but in our previous discussions, thinking about the glycosylation patterns. So when you process the GMP, the amount of sugar residues that are maintained would have variable outcomes as a prebiotic source. So maybe digging deeper into some of that, the manufacturing and what the final product is and what that means for metabolism and function. I don't know if that's a good answer for now, but I'm admittedly learning as much as I can about these protein sources and bugging John Lucy. Thanks, John. So why DIH? One answer is I'm very curious. I've always been curious, and I went into research purely because of that curiosity and wanting to do it for a living. And so you can see we have funding from a number of different sources, some cancer, some infection, some metabolism, and that was a result of being broadly curious. I'm also a Wisconsin native, and the dairy state is home, and thinking about not just commercialization opportunities, but taking things from a starting product to something useful for people. is a motivator for me in that research. And I've been a part of a couple entrepreneurial, taking something we found in the lab and forming a company around it. Just the process of taking raw materials, foods, and in this case milk, and finding new and useful ways to use them better. That's a driver. Any other questions? You guys are hungry?
So you'd mentioned colostrum.
Yes.
So I'm seeing more and more advertisements on colostrum supplements for humans, I believe with regard to suppressing seasonal allergies and those kind of things. I'm assuming that's bovine colostrum, is that correct?
Yeah.
And then what would be the other applications for, you just briefly mentioned colostrum, what are you interested in with regard to colostrum effects on human nutrition?
Well, so initially when I designed some of these studies, I said colostrum, but I'm reminded by my colleagues in dairy science that the colostrum is extremely important for the calf. So we don't want to divert streams of colostrum into human consumption. But it is true that it's picking up in the marketplace. Various companies, Stronger and Parenteral, are marketing this, making milkshakes and sports nutrition drinks out of colostrums. But I think raw milk and even mature milk still has appreciable amounts of many of these things. So you don't necessarily have to steal the colostrum from the calf to get immunoglobulins or lactoferrin or the proteins. So that's kind of my thinking. It doesn't necessarily have to be colostrum. You can just use mature milk and enrich the pieces we need. All right, well, thank you. Heather?
All right. Well, let's thank Joe again and hope